Hi everyone. Okay. Right. In the morning we are discussing about the roles of two promoter hormones, right? So one is auxin, another one is gibberellin. So what we have discussed regarding the auxin, that is what? Root initiator in the stem cuttings. Okay. Then what are the main roles we have discussed? That is, it initiates, no, it promotes flowering in pineapple, not initiating, okay, it is promoting the flowering in pineapple. Then apical dominance, it prevents the obsession in young leaves and flowers, and it prevents the early falling, what is the one, and then it is promoting the senescence in old leaves, that is means by forming the obsession, clear. Then mainly it is inducing the parthenocarpy, the virgin fruit forming, that is in the tomato fruits. Right, so this is what we have seen, and even in the tea plantations we are using our edge making, that is what a gardening process, right? So, and also the main role is of the 2,4-D, that is also along with the 2,4,5-T. These two are also used as a weedicides or herbicides, especially they shows their effect in the dicots than the mature monocots, because monocots are having their degrading mechanisms. This is what about regarding the auxin, whereas the gibberellins. So gibberellin is called what? It is a dormancy breaking hormone, what we call, because it shows antagonistic property with respect to of abscisic acid. Antagonistic means it shows opposite action with respect of the seed dormancy. Because abscisic acid initiates the dormancy, it breaks the dormancy by making alpha amylase synthesis in barley seeds. Amylase, why? Because barley is a starchy seed. If there are some other fatty seeds, there is a lipase activity. If there are some protein-rich seeds like the grams, there will be a protease activity. That is what the amylase or protease or lipase, what I said, that is increased by the activity of the gibberellin. And likewise, it is enhancing the shoot length, right? So along with that, mainly it is increasing the internodal space, not only in the monocots or not only in the zygotes, but increase the internodal space, especially it is prior to the flowering, means called bolting. That bolting process is enhanced especially in the leafy vegetables, and also increasing the length of the stem, that is especially helpful in the sugarcane plants, because by the application or the spraying of gibberellin, we are increasing the yield of sugar only by increasing its length. Agare, what is the application of gibberellic acid spraying andre directly sugar yield increase agate anta alla. First it is increasing what? The length of the stem by increasing the internodal space and accumulates the carbohydrate in the form of sugars. Hence it is increasing the yield of sugar only later again. So first thing andre, it is increasing the internodal space antane. So internodal space is increased, then it is yielding is increased. That is what? 20 tons per acre. So along with that, it is delaying the senescence. That means it maintains the fruit to be there in the plant body. Abscission means what? It makes the detachment, falling off, it detaches from the parent body, means here it is preventing that one. Prolonged delay means prolonged process of aging is maintaining, that means it delays the process of aging and maintains the fruit to be retained itself in the plant body only to get a good marketing value. When there is a market value, because at a time all the vegetables or whatever the particular plant species is available in the market, there will be a rate is decreased. When there is a scarcity, we will get what? A good value for the particular crop plants. Alright, that is what? To get a good value in the market at the time of when the market value is more, we can get that fruit to be available in the market or that vegetable to be available in the market based on the delaying process by applying this gibberellin. This is what the application, right? So this is all about some of the applications regarding the gibberellin. Right? Then coming to another one important hormone which is regulating the plant growth, but it is of basic nature. Right? So that is cytokinin. Okay. Cyto refers to what? Cell. Kinin refers to divide. So here it is a cell dividing hormone, right? How it is identified? So initially in the tissue culture experiments, by Miller and Skug. They were working on a tobacco plant. Okay, so they have taken a tobacco plant. From that, the central region, that is what? Pith. So from the pith, the explant has taken. Along that, they have taken the food and some other nutrients. But here what they added, to increase the growth and to get a plant body or a saplings, they added a hormone called auxin. But along with that, to increase the rate and to get a more easily the shoot or a root, they added the yeast extract. And they thought that there may be the, or they also added the coconut milk or the extract from vasculature or the internodal extract. So the extract from the internodes of the stem or the extract from the coconut milk or the yeast extract. That means we know that yeast is easily get divided. It is a unicellular one, right? And the likewise the coconut milk is having the easily dividing capacity. Likewise the internodal space or a vascular tissues there easily get divided in the plant tissues. That's why the dividing capacity is there, means the extract may contain the property of increasing the division rate in the cells. In that concept, they added along with the agar as a main food material and the nutrients, they added auxin with the combination of these yeast extract or maybe the coconut milk extract or the internodal extracts or vasculature extracts. Then they came to know that this is nothing but the kinetin. Okay, but kinetin is produced by what? That is produced by herring, sperm, DNA, which has been autoclaved. So this we can take as an what? The pith culture of tobacco can be taken as a bioassay. The likewise it has been discovered, the same way we can take it as a bioassay. But here the kinetin, which is the first form of cytokinin which was been isolated and extracted, but it is what? Herring sperm DNA. Herring refers to what here? It is a fish. Herring is a fish. From that fish sperm, the DNA was extracted by autoclaving. What is autoclaving? Just remember the cooker. How the food will be prepared? By the steam. That is what? Autoclaving means the heating with the steam. They are heated the sperm DNA under steam. That is what? The autoclaved herring sperm DNA gave the kinetin. This kinetin is nothing but N6 porphyryl amino purine. But this is what? Not a plant source. It is a non-plant source, because herring, that is a fish, that is animal sourced hormone. Kinetin is an animal sourced hormone. Then later they came to know that the plant source hormones to be where they can be increasing the cell rate, means by the division. That is what? In the Zea mays. Then in the Zea mays they extracted the kinetin property hormones called the name zeatin. What is that zeatin? It is a benzyl aminopurine. In the tissue culture of the biotechnology or strategies, we have studied that BAP. It is used in the tissue culture to initiate the shoot system. So that is what? The cytokinin, which was first extracted on a plant source, is zeatin. Zea refers to what? The species, so the genus name of the plant source which from it is isolated. ಯಾವ ಪ್ಲಾಂಟ್ ಸೋರ್ಸಿಂದ ಇದನ್ನು ಎಕ್ಸ್ಟ್ರಾಕ್ಟ್ ಮಾಡಿದ್ದೀವಿ ಅನ್ನೋದನ್ನು ಇಂಡಿಕೇಟ್ ಮಾಡಲಿಕ್ಕೆ ಜಿಯಾಟಿನ್ ಅನ್ನೋ ನೇಮನ್ನು ಕೊಟ್ವಿ. ಸೊ ಕೈನೆಟಿನ್ ಈಸ್ ಅನಿಮಲ್ ಸೋರ್ಸ್ಡ್ cytokinin. Zeatin is a plant sourced cytokinin. So kinetin is produced or extracted from what? Herring sperm DNA which was been autoclaved. Zea maize, which was produced from the maize corn kernels. ನಾರ್ಮಲಿ ನಾವು ಮೇಸ್ ಸೀಡ್ಸ್ ಅಂತ ಕರಿತೀವಿ, ಅದನ್ನು ಕರ್ನಲ್ಸ್ ಅಂತ ರೆಫರ್ ಮಾಡ್ತಾ ಇದೀವಿ ಅಷ್ಟೇ. ಓಕೆ, ಸೊ ಮೇಸ್ ಕಾರ್ನಲ್ಸ್ ಅಥವಾ ಮೇಸ್ ಸೀಡ್ ಕರ್ನಲ್ಸ್. ಸೊ ಫ್ರಮ್ ದಟ್ ದ ಜಿಯಾಟಿನ್ ಈಸ್ ಪ್ರೊಡ್ಯೂಸ್ಡ್. The chemical nature, that is a benzyl amino purine. So likewise the cytokinin is also invented to extract from the coconut milk and also from the yeast extract. That is all commonly we known as zeatin only, because they are all showing the same chemical nature. Okay, likewise IPA, isopentenyl amine, that is also isopentenyl aminopurine. So this is also one of the cytokinin. So likewise the naturally synthesizing hormones in the animal source and also in the plant source have been isolated. That is what the discovery of this one. So if they ask bioassay, what is that bioassay? That is, the bioassay is tobacco pith culture. Okay, that is one bioassay. Another one is delays senescence, is delaying senescence in leaf, is another bioassay. This is extra. Okay, you have to go through. Clear? So now we know that how the kinetin or the cell dividing hormone called cytokinin has been discovered by the tissue culture experiments of tobacco by Miller and Skoog. Okay, then it has been isolated from an animal source called a herring sperm DNA, which has been autoclaved, that is kinetin. But the first hormone which is isolated under yav baridiri, which one you will write? Kinetin only. But if they ask with specifically by the plant source, the first cytokinin which was a plant source based, that is zeatin. Name the source for the first plant source which was available for the isolation of cytokinin, that is zea maize or maize kernel. Zea maize is a scientific name of maize. Okay, from the kernels of zea maize or maize kernels. Okay, that is zeatin. If they have not given zeatin, they have given BAP, that is benzyl amino purine. That is a chemical nature of zeatin. There is a common name. Zeatin is a common name. The chemical name is benzyl amino purine. Clear? So likewise its isolation is done. Clear? Is short agide na? So then what is the precursor molecule? As I said, this is a basic hormone, right? It is derived from adenine. That is a modified adenine, because adenine is what? It is a purine. Hence we can extract it from tRNA and also from DNA, right? Because both the DNA and RNA, they contains what the purine called? Adenine. That is a common in both, right? Hence we can isolate that from the tRNA and also from the DNA. That is a precursor molecule. From that we can synthesize that one. So this is about their discovery, their bioassay method and their precursor. Where we can see the synthesis, the site of synthesis is the actively growing regions. Where actively growing regions we can see? It is in the apical regions, or it may be the lateral regions. Maybe we can see the buds, the growing bud regions. Even that may be the lateral buds or may be the apical buds. That's why the lateral or apical both. Okay, so in the root tips or in the wherever meristem is present, we can see the presence of this one, that is cytokinin. Clear? What is the role? The first and foremost we can say the role of this one is to synthesize or to produce leaves and also it is helpful in production of the chloroplast in the cells, because we know that chloroplasts are having their own DNA, easily they will get divided, hence they needs to be get divided and synthesis the chlorophyll pigment to be equally distributed, hence to maintain the chloroplast in the cell they are needed. That is what the cytokinin, chloroplast in the cell, they are all produced by the role of cytokinin. First thing. Then it is showing the opposite action to the auxin. That is what auxin is showing, what? Apical dominance. So to overcome the apical dominance, we are using what? This cytokinin. It overcomes apical dominance. What is that one? It will not allow the only apical bud to grow. It will enhances the lateral bud to grow more. So lateral buds are increased. By overcoming apical dominance, that means the lateral buds grows more due to the activity of this one. Hence, what it will form? So it is enhancing the formation of, it is involves in adventitious shoot formation. What is that one? Adventitious means what? Here the shoot is producing, not root. Okay, the shoot is produced other than the plumule, then it is adventitious. That is what here we have taken the pith region. That is not from the plumule, but it is the concentration is varies. If the auxin concentration is less, cytokinin concentration is more, it will give rise to the shoot first. Right? That is what, other than the plumule, the shoot system is developed, hence the adventitious shoot formation we can say here. Clear? The lateral shoots are more. Okay? Then along with that, it is while the formation of morphogenesis, that is what, in the tissue culture, the plant propagation methods, auxin concentration is equal to cytokinin, it will give rise to what? The undifferentiated mass of cells called callus. In the tissue culture media, ide media na tagondiddivi, we see the same setup. The tissue culture media is having explant. Okay, this is explant. Along with the explant on the media, nutrient constituents and auxin and any of the cytokinin form, BAP or IPA, whatever. But the concentration, when they are equally taken, that gives rise to callus. But when the auxin concentration is lesser than the cytokinin concentration, yes, the cytokinin is giving rise to what? Shoot. Hence the shoot develops first and it initiates the shoot formation first. But we need root formation. While discussing auxin, we shared that it forms what? Adventitious roots, because the auxin concentration has taken more than the cytokinin concentration. That leads to formation of a structure, that is a root, that is initiated first. The root develops first. Clear? And mainly it is helpful in delaying senescence. How it delays senescence? It delays senescence by nutrient mobilization, by enhancing the mobilization of nutrients. Mobilization means what? Supplying from the root to the shoot systems, right? So that's why the mobilization of nutrients, or if they are organic, then the food from source to sink. So both in ionic form or maybe in the organic form, right? So they can be mobilized with the more concentration of this cytokinin. That delays the process, because if nutrients are continuously supplied, food materials are completely supplied, So then continuous supplying of these food and nutrients will enhance the growth. They will not become dead, or they will not enter into the senescence phase, that is procrastinating the process. Hence they will get more grow, hence that is delays senescence. So likewise, by producing the leaves and chloroplast in cell and overcoming the apical dominance, adventitious shoot formation or the lateral shoot formations, along with that in the different concentration in the tissue culture medias, we can prepare the desired structure. That is what? It is helpful in the morphogenesis of the plant body. Okay, then this is all about their role, physiological role, and we are using this one in a horticulture methods also, or tissue culture methods also. Clear? So this is all about one of the basic hormone in the plant growth regulators. Clear? Okay. Coming to another one very important and most exclusively used hormone. Bala hormone, out of the 5, the most used prominent hormone which is used in the agriculture purpose is ethylene. Especially in the horticulture departments, without ethylene there will be no proper development of the fruits, because ethylene is known as fruiting hormone. How this was discovered? The discovery of ethylene is very interesting, because as in the Germans, the roads side trees were shedding their leaves very early. Why? Due to the light fluctuations, the street lights which are fluttering based on the light alterations and light inducing mechanisms, they are making the senescence of the leaves. Easily the early falling of the leaves will be observed. But the thing is, they are making the senescence process more easy, right? And later they have observed the orange fruit which was kept as a ripened orange fruit which was kept near to the unripened banana. Okay, the orange, this is what? A ripened one. The ripened orange which was kept near with the unripened banana. By what? The scientist named cousin. Okay, the scientist named cousin who kept the ripened orange and with the near to the unripe banana. What he found after some time, means after some duration, the unripened banana were ripened than the normal period. They are early ripened. How? Because of the volatile compound which was released by this ripened oranges. The volatile compound means easily we can't make them to be characterized easily. We can't collect that one. That was the volatile compound which is produced in the ripened fruits. They came to know that later. That's what the ripened orange made the discovery of this volatile compound, that is a gaseous hormone. Like that, the cousin and others. Itara barra artheno scientist name nenpagtilla, others anta artha. Et al andre, that is what there are many scientists or many people who worked on this anta artha. Et al andre, that is other scientists are there anta. But mainly the cousin and others andre main author yaru irtare avaru hesaru baradini. So cousin and others worked on this volatile compound and they isolated that. That is what the gaseous content, that is C2H4, nothing but ethylene. This was its thing about discovery. But how we can synthesize this one, which is a precursor molecule? Which is a precursor molecule? It is a methionine derivative. Okay, how can we do the bioassay? By keeping ripened fruits with the near to unripened one, especially banana or apple, the unripened fruits, that is a making their process of ripening more faster. They are hastening process, that is speeding up, right? So that is what it's bioassay. Clear? But naturally, as it is available in the form of what? Gaseous, but it can also be synthesized in the form of aqueous solution. Aqueous form, that is ethanophone. So ethanophone is a aqueous form of ethylene. Ethanophone ಅಲ್ಲ ಅದು, ethanophone. Clear? So coming to the roles. How the bioassay was done? One thing, ripening anta helide. Another thing is peanut seeds. The germination of peanut seed was done by using the ethylene spray. The spraying the ethylene on peanut seeds. So what as the result? What will be the result anta nodidre? That has enhanced the lateral growth of shoot apex. Horizontal growth. It is not promoting what? Vertical growth. It is promoting the lateral or horizontal, that is the transversal growth. So transversal growth is observed. It is showing the horizontal or the transverse growth of shoot apex. That is what? This one. Okay. Then second thing here, that was forming the hooks by forming what? Curving. While studying the bean seed also, we will see the hypocotyl is what? Bended, that is curved. The same way in the germination process, it will form a curved hypocotyl that will be appearing. The seedlings are having the shoot apex which is bent like a hook, hence it forms hooks. And third one, it is not only forming the horizontal growth and also forming the hooks, it is also making what? Just compare here. What is the difference here and here? They have been swollen. The axis is enhanced. The germination, the seed axis, which has the shoot axis which is present here, which is initially it is not going vertical, it is growing horizontal. Arshaya, it is growing transverse growth. What I said, that it means it is preventing the vertical growth. First thing. Second thing, the same seed which was applied with this peanut seed, which was applied with the spray of ethylene, that was showing the hook formation, that is a bending of its shoot apex. Clear? Then another thing is, the third result was, it is enhancing its axis by swollen, the bulging one. These were the results, and this is we can consider as a bioassay. And in NCERT they have mentioned the first function as it enhances the horizontal or transverse growth by forming the hooks, by increasing the swollen axis. That is what the three functions we can see. So agar peanut seed germination by spraying the ethylene is its bioassay. Clear? Artha agtidya? So take it as bioassay. In some, after this line, there are some 4 lines after they have mentioned the peanut seed germination by breaking its dormancy. That is what this one. So that's why you can take this as a bioassay of ethylene. By spraying that one, they have observed the lateral growth or horizontal growth or transverse growth in the shoot axis, and then hook formation, and then swollen axis. Clear? These results are noted. So agare first thing idaitu. Then what about the other functions? As we mentioned, it is a volatile one, the gaseous form, which is called as a fruiting hormone, right? So it is a fruiting hormone means it enhances what? Or it hastens fruit ripening rate. How? The gaseous hormone which suddenly enhances the, hikes the respiratory rate in what? The fruits called climacteric fruit. Means when the fruit is developed completely, that will sudden increases its respiratory rate. The sudden hike, not slow hike, the sudden hike in their respiratory rate. That is what such fruits are called, climacteric, okay, fruits. That means here what happened, the sudden hike or rise in what, respiratory rate in the fruits. Okay, this is called what, respiratory climatic. Very important. The respiratory climatic is the activity shown by what, the ethylene hormone. Okay, that means in the fruits where we can see the sudden hike in their respiratory rate, they are called climacteric fruits. That process is called respiratory climatic. Clear? By that it enhances the release of carbon dioxide, and also that means it is breaking that one, and also it is fastening the rate of fruit ripening. This is the major function of ethylene. That is natural form, right? Then along with these two, it is promotes what, senescence. It promotes senescence of what, leaves and fruits. But ಇಲ್ಲಿ ಏನು ಮಾತಾಡಿದ್ವಿ, it produces leaves and shoots and it delays senescence ಅಂತ ಬರ್ದ್ವಿ, right? So here it is enhancing the fruit ripening rate. That effect is called what, respiratory climatic. And here the delaying senescence by mobilization of nutrient is called Richmond-Lang effect. Take it down as note. Richmond-Lang effect. It is also its ಏನು ಹೇಳಿದೆ, ಬಯೋಅಸ್ಸೆಲ್ ಹೇಳಿದೆ, senescence of leaf ಅಂತ, right? So Richmond-Lang effect is showing one of the bioassay method and it is also its function. Clear? So delaying senescence by mobilization of nutrients is called Richmond-Lang effect. That was shown by which hormone? Cytokinin. The respiratory climatic is shown by which hormone? Clear? Okay. So it promotes senescence of leaves and fruits. Along with that, there is a clear mention that it initiates flowering and fruit synchronization in most of the fruits also, but mainly they have mentioned with the example of pineapple. In the early we have studied that the pineapple fruit is, means the pineapple flowering is promoted by auxin. There they mentioned promoted, but here we are mentioning initiated. Clear? The flowering in the pineapple is initiated by what? Promoted by means auxin, initiated by means ethylene. Clearly they have mentioned it initiates flowering and synchronized fruiting in pineapple fruits. Okay, it also useful for flowering and synchronized fruiting of many fruits, but they have mentioned with a specific example of pineapple. But the same pineapple example we have quoted in auxin, but for promoting the flowering process. Clear? Okay. Then along with that, the internodal space is enhanced, okay, but it is in the waterlogging condition where the plant body is, see, if it is a water level, it is a soil, The water level is up to this. This is the soil. The plants, they are monocotyledon, especially I am saying with respect to of paddy. Okay, because we know that paddy fields are any time before the seed setting, means till the complete seed maturation, they require waterlogging condition. Now ನೀರು ಕಟ್ಟಲೇಬೇಕು, ಭತ್ತ ಬೆಳಿಬೇಕು ಅಂದರೆ ಮೇಜಿಗೆ ಹಾಯಿಸಿದಂಗೆ ಬಿಟ್ಟು ಬರಲಿಕ್ಕಾಗಲ್ಲ. ದಟ್ಸ್ ವೈ ಹಿಯರ್ ದ ವಾಟರ್ ಲಾಗ್ಡ್ ಕಂಡೀಷನ್ ವೇರ್ ದ ಪ್ಲಾಂಟ್ ಬಾಡೀಸ್ internodes and lower nodes are submerged in the water condition. The leaves may be get submerged and they may leads to the wilting by blocking the respiratory pathways. Hence, they enhances the internodal spaces. What happens now? Earlier I have shown how many nodes? 3 nodes, but they were up to this one. But now the internodal space is what? Increased. And you can see the first node or the lower node which is near water, the surface of water, but it is not now submerged. That means what? The water level, above the water level, the leaf will be gets exposed. There will be no wilting. So especially in the fields where water is logged, okay, that means the submerged water plants are enhanced their internodal space by the effect of this ethylene, that is the rice field or nothing but the paddy fields, to avoid wilting or submerging submerged leaf conditions to take above the water surface. Ethylene is important. Clear? So it makes the plant body to enhance its internodes in the waterlogged or submerged conditions and makes the leaves to extend and above the water surface. Clear? Submerged level ginta higher level ge tagombarutte. So this is in the rice or the what we call the paddy plants. Clear? It is one typical example they have quoted. Clear? And mainly it is leading to the formation of feminized condition in the fruits, means feminized condition of the flowering. What is feminine character? That is developing on the female flowers, where we can see the female flowers, where the female flower is separate, male flower is separate in the plant body, right? So they are bisexual, okay, and the thing is the main plant body which may bear the male flower and female flower separately, or it may bear only male or only the female in the one plant body. That is what we have seen the gytonogamy, right? So here in that means it enhances or brings feminism or feminine condition in what? Flowers. That means the female flowers are produced more. Where? In Cucurbitaceae family. What is the Cucurbitaceae family example? In NCERT they have given with a cucumber, but we can add it as a watermelon also. Maize is also monoecious. Cucurbitaceae, maize, cara, cucurbita, they are all what? Monoecious, but bears male and female and the separate. The flowers are separate. Unisexual flower, bisexual plant. Gytonogamy, that's what I mentioned. Don't get confused. Okay? So it brings what? Feminine flower. That means it develops the female flowers. Why? What is the need of this one? What is the advantage? Yes, what is getting the female flowers benefit? More yield. Yes, what is the yield by the flower? Seed. How the seed will get developed in the angiosperms? Gynoecium is nothing but the carpels, right? They are the deciding factor, the nature and the type of fruit it will develop. That means if there is no gynoecium, even though we are having the plenty of male flowers, there is no use, because without a gynoecium there will be no fruit, because ovary is present in the gynoecium itself. Hence, to enhance the number of fruits in the plant body, the carpel rich condition, that is the feminine character, is brought by spraying these hormones. Naturally ಆಗ್ತದೆ, and even we can use from the ethaphone also. Next ಅದನ್ನು ಮಾತ್ರ ಲಾಸ್ಟ್ 3 ಎಫೆಕ್ಟ್ಸ್ ಇದೆ ಅದಕ್ಕೆ. ಓಕೆ, ಸೊ ಇಷ್ಟು ಕ್ಲಿಯರ್ ಇದೆಯಾ? ಸಿ ದ ethylene in the natural form, which is producing the lateral or transverse growth of the shoot axis by seed germination, or it gives the hook formation, or it swells the axis. This is one thing. Second thing, it fastens the rate of fruit ripening by increasing its respiratory rate. Clear? That respiratory rate is suddenly hiked. That effect is called respiratory climactic. This will be observed only in the fruits where at the time of maturity they are increasing their respiratory rate. Those fruits are called climacteric fruits. Clear? Okay. Then it promotes senescence of the leaves and fruits. It initiates flowering and fruit synchronization. Why we are using this one, synchronization? Say, if the flowering is alternated or even the fruiting is alternated in the different periods, the harvesting process makes to be harvesting process will be tedious. It requires more workers. It requires more investment. It is not economic friendly. Every time we need to pluck the fruits means we need more workers. That means we will not get a economic friendly conditions in the fruit developments. Andre namge adu value illa. Clear? Hence, the synchronizer means at a time we can collect the fruits and we can get into the market. One thing. So when there is a rate, we can make them. We know already there is one hormone which can delay that one. Right? So when the market value is there, we can spray this one, we can get the synchronizer condition, we will get the market value. That is what the application of this one. Hence, I said that this plant growth regulator is most extensively used in the agriculture purposes than any other. This is only the reason, to get the female flowers, to get the more fruits, and to get this one. Varun, clear? So this is all about what? Ethylene, that is in natural form. If we can take that in a synthetic form, that is ethaphone, the spraying of this one, which will brings what? One is female flowers are more developed in the Cucurbitaceae family. That is one of the major role of ethaphone. Okay, along with that there are two more benefits, two more application of ethaphone. Yes, so ethaphone brings what? What is the application? It is the aqueous hormone, what I said. In the aqueous condition, the synthetic hormone is available. What are the uses? So first one is, we will get the feminism or female flowers in Cucurbitaceae. Okay. ಬಟ್ ಕನ್ಫ್ಯೂಸ್ ಆಗೋದು ಬೇಡ. ಒಂದು ಮೇಲ್ ಫ್ಲವರ್ಸ್ ಮಾತ್ರ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡೋದು ಅಂದರೆ ದಟ್ ಈಸ್ ಗಿಬರ್ಲಿಕ್ ಆಸಿಡ್. ಓನ್ಲಿ ಇಟ್ ಮೇಕ್ಸ್ ದ ಮೇಲ್ ಫ್ಲವರ್ ಪ್ರೊಡಕ್ಷನ್. ಓಕೆ, ಅದು ಎಕ್ಸ್ಟ್ರಾ ಬಟ್ 10 ಪಿರ್ಲಿ ಅಷ್ಟೇ. ಓಕೆ, ನೆಕ್ಸ್ಟ್ ಅಲಾಂಗ್ ವಿತ್ ದಟ್ ಎನಿ ಅದರ್ 2 ಫಂಕ್ಷನ್ಸ್ ಮತ್ತೇನಿದೆ? ಇಟ್ ಈಸ್ ಎಸ್ಟನಿಂಗ್ ವಾಟ್? ಫ್ರೂಟ್ ರೈಪನಿಂಗ್. ಓಕೆ, ಸ್ಪೀಡ್ ಆಫ್ ದ ಪ್ರೊಸೆಸ್ ಆಫ್ ripening. Okay, it hastens fruit ripening. And last one, yes, ಒಬ್ಬರು ಏನು ಹೇಳ್ತೀರಿ, ಇನ್ನೊಬ್ರು ಏನು ಹೇಳ್ತೀರಿ? ಎಸ್, it accelerates senescence, nothing but by brings the abscission formation. That means it makes the plant body to get detached with that new fruit which has been developed. Abscission is accelerated. Clear? So this is all about the role of natural and also synthetic ethylene forms. ಕ್ಲಿಯರ್? ಗೊತ್ತಾಗಿದೆ ಎಷ್ಟು? ಓಕೆ. ಸೊ ಇಲ್ಲಿ ತನಕ ನೋಡಿದಂಥದ್ದು ಪ್ರಮೋಟರ್ಸ್ ಆಯಿತು. ಆದರೆ ಈ ಹಾರ್ಮೋನ್ ದಿಸ್ ಒನ್ ಈಸ್ ವೆರಿ ಸ್ಪೆಷಲ್ ಬಿಕಾಸ್ ಆ್ಯಸ್ ಐ ಸೆಡ್ ದಿಸ್ 3 effect, that is it is showing triple effect. In the previous that has been asked, the triple effect showing hormone is ethylene. What is the triple effect? One is transverse growth of the shoot axis, other one is hook formation, third one is Solon axis. These are the three we wrote. These are the three effect which were brought in the peanut seed germination by the activity of ethylene spray. Clear? That is in the natural form. Hence this is called the triple effect showing hormone. Make a note. ನಾನು ಯಾವುದು ಇದುವರೆಗೂ ಎಕ್ಸ್ಟ್ರಾ ಹೇಳಿಲ್ಲ, ಇರುವಂಥದ್ದನ್ನು ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಿದ್ದೀನಿ ಅಷ್ಟೇ. ಹೆಂಗೆ ಕೇಳಿದ್ರು ಆನ್ಸರ್ ಮಾಡಬೇಕು. ಕ್ಲಿಯರಾ? ಯಾಕೆಂದರೆ ಬೇರೆ ಎಲ್ಲ ಚಾಪ್ಟರ್ಗಳನ್ನು ನಾವು ಇದನ್ನೇ ಮಾತಾಡಿದ್ದೀವಿ. ಸೊ ರೆಸ್ಪಿರೇಟರಿ ಕ್ಲೈಮ್ಯಾಟಿಕ್ ಅಂದರೆ ರೆಸ್ಪಿರೇಟರಿ ರೇಟ್ ಹೆಂಗೆ ಇನ್ಕ್ರೀಸ್ ಆಗುತ್ತೆ, ಬೈ ರಿಲೀಸಿಂಗ್ ಕಾರ್ಬನ್ ಡೈಆಕ್ಸೈಡ್ ನಮ್ಗೆ ಗೊತ್ತಿದೆ. ದಟ್ಸ್ ಇಟ್. ಹಾಗಾದ್ರೆ ಇಲ್ಲಿ ಅದ್ರ ಜೊತೆಗೆ ಇಟ್ ರಿಲೀಸಸ್ ಕಾರ್ಬನ್ ಡೈಆಕ್ಸೈಡ್, ಎಸ್ ಅದಿರಬೇಕು. ಓಕೆ. ದ ಇಥಿಲಿನ್ ಮೇಕ್ಸ್ ದ ರಿಲೀಸ್ ಆಫ್ ಕಾರ್ಬನ್ ಡೈಆಕ್ಸೈಡ್ ಅಂದರೆ ಇಟ್ ರಿಲೀಸಸ್ ವಾಟ್ ಬೈ ಬ್ರೇಕಿಂಗ್ ದ ಕಾರ್ಬೋಹೈಡ್ರೇಟ್ ಆರ್ ವಾಟ್ ಎವರ್ ದ ಫ್ರೂಟ್ ರಿಚ್ ಶುಗರ್ಸ್, they are broken down in the process of ripening. Hence the completely ripened fruit is not suggested to eat. Will we eat the completely ripened fruit? Yes, Kushi only eat that one. The completely ripened fruit will Deepika will also eat. Why? Means they are wine lovers, because the completely ripened fruit is always having the almost at the stage of it is entering to the rottening. ಕನ್ನಡದಲ್ಲಿ ಹೇಳಬೇಕಂದ್ರೆ ಕೊಳತು ನಾರಕ್ಕೆ ಸ್ಟಾರ್ಟ್ ಆಗುತ್ತಲ್ಲ ಅವಾಗ ತಿನ್ನೋಕ್ಕೆ ಅಂತ. ಬಾಳೆಹಣ್ಣು ಫುಲ್ ಕಪ್ಪಗಾಗಿರುತ್ತೆ, ಔಟರ್ ಮೋಸ್ಟ್ ಪೆರಿಕಾರ್ಪ್, ತಿಂತೀರಾ? ಫುಲ್ ಮೆತ್ತಗಾಗಿರುತ್ತೆ, ತಿಂತೀರಾ? ಇಲ್ಲ ಅಲ್ಲ, ಒಳಗೆ ಚೆನ್ನಾಗೇ ಇರುತ್ತಂತೆ ನೋಡ್ರಿ. ಹಾಂ ಇಲ್ಲ. ಓಕೆ, ಈಗ ನೀವು ಅದನ್ನ ನೆನ್ಪು ಮಾಡಿದ್ದು ಒಳ್ಳೆದಾಯಿತು. ಈಗ thinning of another one process, thinning of pericarp of walnut, cotton, cherry. ಎಲ್ಲಿ ಇದರ ಎಫೆಕ್ಟೇ ಬರ್ಗಡ್. ಯಾರ್ದಾರು ರೂಮಲ್ಲಿ ಅಂಟ್ಕೊಳ್ತಾ ಹೋದ್ರೆ ದಯವಿಟ್ಟು ಹೊಸಿಬೇಡ್ರಿ, 2 ಜೀವಕ್ಕೆ ಊಟ ಕೊಡ್ರಿ. ಥಿನ್ನಿಂಗ್ ಆಫ್ ವಾಲ್ನಟ್, ಚೆರ್ರಿ ಆ್ಯಂಡ್ ಲಾಕ್ಡೌನ್ ಬೇರೆ, ಕಾಟನ್. ಕ್ಲಿಯರ್? ದೆನ್ ಅರ್ಥ ಆಯ್ತಾ? ಇಟ್ ಲಿಟ್ಸ್ ದ ಎಷ್ಟರಿಂದ ಫ್ರೂಟ್ ರೈಪನಿಂಗ್ ಅಂತ ಮೆನ್ಷನ್ ಮಾಡಿದ್ವಿ. 2 ಎಕ್ಸಾಂಪಲ್ ಕೊಟ್ಟಿದ್ದಾರೆ, ಆಪಲ್ ಆ್ಯಂಡ್ ಟೊಮ್ಯಾಟೊ. ಅವು ಗೊತ್ತಿರಲಿ. NCERT ಏನು ಎಕ್ಸಾಂಪಲ್ ಕೊಟ್ಟಿದ್ದಾರೆ ಅದು ಮಸ್ಟ್ ಆ್ಯಂಡ್ ಶುಡ್ ಯು ಶುಡ್ ರಿಮೆಂಬರ್. ಸೊ ಟೊಮ್ಯಾಟೊ, ಆಪಲ್, ಸರ್ ನಾವೊಂದು ಇಡ್ತೀವಿ ಅಂದರೆ ಆಯಿತು ಬರ್ಕೊಳ್ರಿ ಅದನ್ನು. ನಾರ್ಮಲಿ ಸೊ ಹೌ ಯು ವಿಲ್ ಡೂ ದ ಬನಾನಾ ರೈಪನಿಂಗ್ ಪ್ರೊಸೆಸ್ ಇನ್ ಹೋಮ್ಸ್ ಕಾಮನ್ಲಿ? ಓಕೆ, ಎಸ್, ಅನದರ್ ಒನ್ ಫಂಕ್ಷನ್, ಇಟ್ ಇನಿಷಿಯೇಟ್ಸ್ ದ ಫ್ಲವರಿಂಗ್ ಇನ್ ಮ್ಯಾಂಗೋ. ಇಟ್ ಇನಿಷಿಯೇಟ್ಸ್ ದ ಫ್ಲವರಿಂಗ್ ಇನ್ ಮ್ಯಾಂಗೋ. ಮ್ಯಾಂಗೋಗೆ ಯಾವುದಕ್ಕೆ ಅಂದರೆ ನ್ಯಾಚುರಲ್ ಫಾರ್ಮಲ್ಲಿ. ಓಕೆ, ದ ಮ್ಯಾಂಗೋ ಫ್ಲವರಿಂಗ್ ಈಸ್ ಇನಿಷಿಯೇಟೆಡ್ ಬೈ ಇಥಿಲಿನ್. ಓಕೆ, ಬಟ್ ಹೌ ಇಟ್ ಈಸ್ ರೈಪಂಡ್? ಕಾಮನ್ ಆಗಿ ಫ್ರಾಗ್ರೆನ್ಸಿ 6 ವಿ ವಿಲ್ ಯೂಸ್ ದ ಫ್ರಾಗ್ರೆನ್ಸಿ 6. ಓಕೆ, ಆರ್ ವಿ ವಿಲ್ ಯೂಸ್ ಅ ಕ್ಲೋಸ್ಡ್ ಎನ್ವಿರಾನ್ಮೆಂಟ್. ವಿ ವಿಲ್ ನಾಟ್ ರಿಪೀಟೆಡ್ಲಿ ಓಪನ್ ದಟ್ ಒನ್, ರೈಟ್? ಆರ್ ದ ಸ್ಟೋನ್ಸ್ ಆಫ್ ದ ethylene, that is ethaphone, they are before making the aqueous solution they are used in the ethaphone form, that is solidified one also. That will release us by coming in contact with the oxygen, it will release the volatile compounds that will make the fruit to be get extend in the rate of respiration, hence that will lead to respiration. That is the ripening one. Clear? Okay, so this is all about ಹಣ್ಣು ಮಾಡೋ ಹಾರ್ಮೋನ್. ಓಕೆ, that is the fruit ripening hormone called ethylene and its synthetic form ethaphone. The last one, and this is also called dual nature hormone. Why? It shows both promoting activity and also inhibiting activity. Clear? Okay, coming to the one and only inhibitor hormone, abscisic acid. Abscisic acid. So we will inhibit up to this level. In the next we will promote this contents. Okay. So abscisic acid, it is also acidic hormone synthesized by the precursor molecule called what? Carotenoid. That is what? The xanthophyll. Exactly. Violoxanthin. Okay. Clear? Right.